Hi, my name is Guilherme Oliveira and I am the head of the Vale Institute of Technology. This video is in support of the BIOSCAN project. We firmly believe that the BIOSCAN project and the international community around it are essential for the production of new tools, knowledge and sharing of information that will help us in describing and protecting the Amazon rainforest. The Vale Institute of Technology is located in the city of Belém, in the state of Pará, in the northeastern Brazilian Amazon. We conduct work in many different biomes in Brazil, but what I'll describe today is conducted mostly at the National Forest of Carajás in the state of Pará in the Brazilian Amazon. The work in this region is especially important because the region has suffered intense pressure from cattle ranching and also because the national forest was designed to host within its boundaries industrial mining activities. One example is the S11D complex. DNA barcodes have been essential to help us describe the biodiversity in the region and, along with other tools, to design better conservation methods. One example was the use of traditional taxonomy methods and DNA barcodes to describe the flora of altitude ferruginous fields in the region. These altitude fields host a very peculiar flora. We produced what is still the first flora of the entire Amazon region and the first flora with reference DNA barcodes. The flora was published in 4 volumes of the National Botanic Gardens journal, and from 200 known species when we started the work, we have now over 1,066 species, 8 of which are restricted to the region. The barcode library now doubles what is known of the flora of Brazil. This mountainous area also contains thousands of caves that occur mostly on the edge between the forest and the canga fields. To reveal the biodiversity that occurs in cave habitats, we have intensely used DNA barcodes, sometimes in addition to genomics and high-resolution morphological tools. This is an example of 2 small species of water beetles that are very difficult to distinguish morphologically. The use of molecular tools have been instrumental in helping us to untangle this and other taxonomic groups. We are also developing reference library for fish so that we can use this information to monitor the water bodies within the region. Specimens are collected along a number of monitoring points in the streams. DNA barcode analysis has revealed a number of species that are still unknown to science. We are now in the process of describing these new species. We have also collected water samples for using eDNA analysis to monitor these water bodies. The results indicate that eDNA is very effective in pointing to the different fish populations at the sampling sites. We have also used malaise traps to capture arthropods and study the evolution of land reclamation in iron ore mine settings. The setup also allowed us to look at the vertebrate population by detecting their DNA in blood-feeding insects. This year, we will set up malaise traps at different agroforest sites throughout Brazil. We expect to capture how the setup of agroforest settings in previously degraded lands will impact arthropod biodiversity. At ITV, we are deeply committed to discovering, monitoring, and conserving nature, especially the Amazon region. We firmly believe that by being connected to BIOSCAN, we will be able to move faster and with much better tools to achieve our goals. Thank you.